goes on the screen. It is absolutely my pleasure today to introduce Dr. Marzi, who is not only an amazing, wonderful scientist, but also a dear colleague of mine. I've been trying to get her to come to UCI and give a talk for a couple of years now, and we're finally doing it via Zoom, of all things. So Dr. Marzi is the chief of the Immunobiology and Molecular Virology Unit, which is part of the Laboratory of Virology. It's the intramural division of NIH. And she is in Hamilton, Montana at the Rocky Mountain Laboratories. She obtained her PhD from the very prestigious, I'm going to butcher the name of this university, Friedrich Alexander University. Close enough.
Yeah.
In Newburgh, Germany, working on cellular attachment factors for filoviruses. And I guess fell in love with filoviruses and actually did a postdoc both at the Canadian Public Health Agency, well, Public Health Agency of Canada, and then moved to Rocky Mountain Labs, where she climbed the ranks to her current position. Dr. Marzi has received a number of awards. I'm not going to rattle them all off because it would just take too long. But I'd like to at least mention the fact that she is one of the NIH Distinguished Scholars, which is not a small feat of accomplishment. So congratulations. And she is leading a very vigorous research program, both looking at vaccines against filoviruses, leveraging the recombinant vesicular stomatitis virus platform for other pathogens. And more recently, like everyone else, she's also pivoted to doing some COVID work, as no self-respecting virologist could not do that this year. And she has a tremendous amount of publications for somebody who just became a lab chief. It's really impressive, Andrea, and has done a lot of community outreach as well as field work in Africa, especially during the 2013 to 2016 Ebola epidemic. So she's not only a fabulous bench researcher, but she's also an experienced field researcher. And she is a reviewer for many prestigious journals, including like New England Journal of Medicine and Lancet. Has a ton of presentations, very highly sought after. And it is absolutely our pleasure to welcome you here today. I and others here are looking very much forward to hearing your talk. So without taking any more of your time, welcome, Andrea, and I'll give you the floor.
Thank you, Ilham, for the super kind introduction. And I'm really happy that we finally made it work, that I could present my data or our data. Some of them were in collaboration with you at UC Irvine. This is really cool. I always, really wanted to come and go to California, particularly this time of year, but, winter in Montana is long, but let's do it via Zoom. I'm super excited to share our data with you. I will talk about Ebola virus and the vaccine against it, but then also, as Ilham mentioned, about our approach of a COVID-19 vaccine, because last year it was like either do that or stay at home. So it was really a no-brainer. All right, I hope you all can see my screen just all right. Perfect. And also, please, if you have questions, feel free to interrupt. I'm not necessarily always a firm believer, in afterwards or things got forgotten. I don't mind if you want to talk, have questions. So as Ilham mentioned, I'm actually located, even though I'm NIID, at the Rocky Mountain Laboratory, which is not where our main campus is. On the East Coast, we are in beautiful Bitterroot Valley in Montana, on the western part, as depicted up here with this story, of the state. And we have the Bitterroot River running through. So this is the campus when this building here was still under construction, and that's actually where I am right now, sitting, looking north out of these windows. And this also houses a big BSL-3 and BSL-4 laboratory. So this is the building close up. You can see the windows are all our office spaces, behind this BSL-2 space, and then BSL-4 and BSL-3. This is what the BSL-4 space looks like, or actually looked like when we first moved in 2008. it was super nice and clean, and it's big. We have like, this is 1 out of 4 suites. So we have the luxury of continue to work even though we have annual shutdown. So we're just always having a suite open, and we just move around whenever it's needed. And we also have a very big animal space, which is really nice to do translational research and not just in vitro stuff, where you can then confirm your data in animal work. And then this is what we look like when we work in there, which, is like, as you have probably seen before, it's actually fun. I miss it when I don't get to go in for a while, because for me, it's me time. There is no phone calls, no email. You can just focus on pipetting or working with animals. The only downside is you have to wear 3 pairs of gloves and you are a little bit bulkier. So getting around is sometimes a little cumbersome when you think you're not quite as big as you think you are. I have here a short movie showing the lab. Let me put this back here. I need to, otherwise the video won't start. So this is showing you again where the Rocky Mountain Laboratories are located, and then it gives you a quick idea of what an entry in the BSL-4 looks like. So this was filmed in 2008, so almost 13 years ago. And this here is the BSL-2 block. This is BSL-4, and it's connected through this hallway. When we go into the BSL-4, we have to change out of our street clothes into scrubs. And then we do a suit check every week. We change the gloves on our suits every week. That has to be done because they are the weakest point, since it's just rubber. And we go through a lot of duct tape, because the gloves are secured to the suit with duct tape. Any leaks, pinholes that you might have are patched with duct tape from inside and outside. As you can see in this video also, we tape our socks to the scrubs with duct tape, and also our gloves eventually are being taped with duct tape to our scrubs. So it's basically a duct tape commercial. We are able to wear headsets in order to communicate with other scientists. And then we are going into the laboratory, and these APR doors, so they are leak proof, and they open with this system. Here is how we work. We work in BSCs just like you would in BSL-2, BSL-3. All the liquid is decontaminated overnight in microchem, which is the yellow stuff in the buckets that you see in the BSCs, and the solid waste, and everything gets autoclaved. So we work in there without restrictions of viruses per suite or so. So as long you only can handle a virus in a BSC at a time, though. The animal space is rather big. We have 7 animal rooms and 2 rodent rooms. So that is super helpful for also doing non-human primate, and Heinz Feldman is doing some pig work. And then, like in all the select agent spaces, the viruses here are under lock and key. The scientists themselves do not have access. We have to request viruses, and they're stored in liquid nitrogen. So we put in emails, and we will receive those. Then we are done. When you're done with your work, you take a chemical shower. It is 3 minutes of chemical and 3 minutes of water rinse. And then you come out, you hang up your suit, you get out of your scrubs, take a personal shower, and then you go back and get dressed. So, even though we work with those dirty bugs and animals, and people always think we are so dirty, we're actually quite clean because we shower a lot. And as I said, it's different to work in there, because you also cannot really forget anything, because there's no media stock inside or anything. What you need to bring. So if you're a person who forgets a lot of things, then at some point you run out of favors from your coworkers, because you have to call them and have them put it in a chemical shower for you or dunk it in. So it's quite educational for some people, I have to say. But nevertheless, so now you have an idea of where we are and how we work. So the science that my lab is particularly doing, and I have been doing since I became a postdoc here, is mainly focused on Ebola virus. Even though now we have approved vaccines and actually therapeutics against this virus, back in the day we did not. So I became interested in it after my PhD work in Germany, where I characterized attachment factors for this virus and then really wanted to work with the real thing. So I was lucky to score a postdoc position with Heinz Feldman in Winnipeg, Canada, to start my work on filoviruses. My work was started with characterizing Ebola virus and then the vaccine further that he had developed together with Tom Geisbert. So Ebola virus is a member of the Filoviridae, or also filoviruses, as you've probably heard, and it's a member of the species Zaire Ebola virus. The virus is enveloped and filamentous and contains a non-segmented negative single-stranded RNA genome. The genome is 19 kilobases in size and encodes for 7 genes, which are translated into 9 proteins. The virus replicates in the cytoplasm of an infected cells, and cell budding occurs on the cytoplasmic, on the cell membrane. Molecular characterization of the virus has revealed that it has host interferon antagonists. They are the proteins VP24 and VP35. And Ilham has actually done some work with Chris Bosler and Tom Geisbert towards that, and I'm sure you've heard about it. My protein of interest for the vaccine, as well as the pathogenicity work that we have done over the years, is the glycoprotein. That is the only viral protein that's expressed on the virus surface. And you can see it here on the surface, on those bubbles of the virus. The genome is depicted here, as you can see. So here is the glycoprotein open reading frame. The virus has been in the news, not just because of the epidemic in 2013 or 2016, but also just lately again, because there is right now an outbreak ongoing in the Democratic Republic of the Congo, and they just came out of an outbreak that lasted for 2 years and was the largest in that country. But also in Guinea, Ebola virus has been identified as causing human disease again, and also some fatalities. So right now, WHO and the Doctors Without Borders are battling Ebola virus on 2 fronts in Africa again. We don't know for sure yet how these outbreaks started, but the regular routes of transmission are usually from either the reservoir or in contact with bushmeat, because the current transmission cycle is believed to be that bats are potentially the reservoir host for Ebola virus, and they can infect non-human primates in the forests, and can also infect humans through direct contact, either by really like contact with a live bat or by consuming bushmeat. The same is true for non-human primates. It can spill over from them into the human population by the same routes. And a lot of work has actually been done in Africa already, trying to educate hunters that a dead non-human primate in the forest is not necessarily a gift from the gods, and you rather be cautious of eating it or not eat it because it could carry a disease. And then, of course, Ebola virus is transmitted human to human by body fluids. Like many other diseases, the disease starts with flu-like symptoms that can progress to severe disease, including mucosal hemorrhages and petechiae. In this picture over here, you can see where which parts of the body or which organ systems are influenced or infected. Early target cells are generally the spleen and the liver, but then the virus really spreads systemically. And we, of course, do not have samples from humans here, but from non-human primates. And there, whenever we look for virus replication in any of the tissue samples taken from a terminally ill animal, we find the virus. And the destruction that the virus is causing in the body and the dysregulated immune response to the infection usually results in multi-organ failure and death. And for Ebola virus, that can be up to 90%, depending on the size of the outbreak. Thankfully, in the epidemic 2013 to 2016, the case fatality rate was only about 60%, so much better than it used to be in the past. We studied a virus in the laboratory in animal models, and we have actually several different options, which is really nice for disease observation as well as countermeasure development, including vaccines and therapeutics. So normally when we have a really cool vaccine approach or an interesting therapeutic, we start in what we call screening models that are either mice or hamsters or guinea pigs. For each of those, we have a rodent-adapted Ebola virus. These Ebola viruses that have been adapted carry mutations, so the rodent interferon antagonistic response is altered, and then they cause disease. So that's actually really cool. However, for many studies, when we then also want to move towards licensing of one of our vaccines or therapeutics, we need to go into a model where we can use a wild-type virus. And generally, that's the macaque, because the macaques are the best animal model that we have so far for actually any of the filoviruses, because they cause disease very similar to humans. There's an interesting difference between rhesus and cynomolgus macaques, where in rhesus macaques, the disease progression is about 2 days delayed, but other than that, the disease itself is the same. So usually for therapeutic studies, we use the rhesus macaque, and for vaccine studies, the cynomolgus macaque, in order to really raise the bar to its highest. My focus has been, since I started to work with Heinz Feldman in Canada, on the Ebola virus vaccines. And as I said, normally we start to work in rodents, then we go into non-human primates. And then if we find something that's very interesting to an industry partner, there might even be clinical trials. For Ebola virus, since it was discovered in 1976, many vaccine approaches have been tried. Some of them were successful only in rodent or in non-human primates, but quite a few actually progressed, particularly during the epidemic, into clinical trials. And these clinical trials were mainly done for what we live attenuated vaccine based on vesicular stomatitis virus VSV-EBOV, an adenovirus 5 based Ebola virus vaccine, or a combination of both, which was developed in Russia called Gamevacambi. And then, of course, which I'm sure you've heard now with the COVID vaccines, chimp adenovirus vectors, or a chimp adenoprime with an MVA boost, Ad26 prime with an MVA boost, or a DNA vaccine. All of these were tested in phase 1, 2, and 3 clinical trials during the epidemic. And people were followed for adverse reactions and, of course, for immunogenicity. And that was determined then via antibody and T-cell responses. And out of all these trials, the world now had in 2017, 2 licensed vaccines in China, which was based on adenovirus 5, and in Russia, the combination between Ad5 and VSV. The VSV Ebola virus vaccine that I will be also be talking about in the next few slides a lot was licensed by the US FDA in December 2019. So that was actually super exciting news for us, because finally, there would have been a vaccine for us to get. So we would be better protected working with this virus. But we never got around to get vaccinated because then COVID hit and nothing happened. So hopefully our occupational medical person will follow up with that and get us vaccinated. So what is a VSV and what is that vaccine? So vesicular stomatitis virus is a rhabdovirus similar to Ebola virus, single-stranded negative RNA. It has an even smaller genome and only 5 proteins. So we have a very good reverse genetic system for it that we can easily manipulate and put in all kinds of proteins that we want to express from a vaccine. So the virus itself actually does exist in the United States. It usually causes very mild infections in livestock. They get blisters, which is not a bad thing. However, the picture to a veterinarian looks very similar, like foot and mouth disease. So that's why VSV has kind of always this bad reputation. However, when humans get infected, there is usually asymptomatic to nothing happening. VSV, as I said, is genetically easy to manipulate, and its glycoprotein manages to enter almost any cell type, and it grows to really high titers in vitro. As any rhabdovirus, VSV has this characteristic bullet shape, and the outside is decorated with its glycoprotein G. Why it is so attractive as a vaccine platform, despite that, it's a live attenuated vector, is because the vaccine vector, when you put in the antigens, gets attenuated. So it doesn't replicate as well as the wild-type virus does. This vaccine is efficacious after a single dose, which is really desirable, particularly in the parts of the world where Ebola virus is a problem. There is very limited global pre-existing immunity in humans. And if there is pre-existing immunity, it's usually directed towards the glycoprotein G, which in a lot of our vaccines is not present. And in the next slide, I'll show you why that is. VSV has also been described to be a very strong and rapid inducer of immune responses. And as I will also show you data later, the vaccine is also effective as a very low dose in non-human primates. And in a very basic way, how we work with this virus is basically we take the virus, its genome, cut out the glycoprotein G information, put in our antigen of interest, in this case, the Ebola virus glycoprotein GP. And what we get after we transfect cells with this vector is a virus that carries the glycoprotein of interest on the surface and replicates. So here is a schematic, a little bit more scientific, how this actually really worked. So Jack Rose's lab in the 90s developed this reverse genetic system. And it's a plasmid that is under the control of the T7 promoter. It has the nucleoprotein, phosphoprotein, matrix, glycoprotein, and L, the RNA-dependent RNA polymerase. Then in this vaccine approach that we call the delta G, we cut out the glycoprotein G information and insert the Ebola virus glycoprotein in its place. And then you transfect cells with this plasmid as well as plasmids expressing the replication complex proteins, the polymerase, the nucleoprotein, and the phosphoprotein. And after a few days, you see starting CPE, like plaques form in your cell culture, and you have recovered VSV, which looks like characteristic bullet shape, like a rhabdovirus, but it carries the Ebola virus glycoprotein on the surface and it replicates. So this actually was developed in the early 2000s by Heinz up in Canada already. However, it wasn't until 2005 that the first paper was published that a single dose of this vaccine of 10 to the 7th PFU protected 28 days prior to lethal challenge. He and Heinz and Tom Geisbert at UTMB then conducted numerous studies characterizing this vaccine further. And at some point, I got involved. And we know now that we can actually blend multiple filovirus or EVSV vectors together to make a multivalent filovirus vaccine. Together with Ilham, we looked more into the mechanism of protection in a study that was published in 2013. And we found that the antibodies specific to the Ebola glycoprotein are really critical for protection and not so much the T-cell response. And then during the epidemic, we wanted to know how fast does this vaccine work? And we found that in non-human primates, this dose of 10 to the 7th PFU protected uniformly within 7 days. And the clinical trial data from Guinea actually confirmed that, where they saw that the vaccine in humans was protective in about 10 days. So that was super exciting because for some scientists from here that went later in the epidemic, they got vaccinated in the trial, and they knew that if they would go to Liberia 2 weeks after the vaccination, they'll be fine. So that was really giving a lot of people peace of mind. So the other question that we didn't know about was, when Merck made this vaccine and the dose was administered as 10 to the 7th PFU, is that really necessary? Could we dilute it 1 to 10 or 1 to 100, and instead of with, like a vial of 10 doses, vaccinate not 10 people, but maybe 100 or even more? So that was a really important study that Heinz and I did also closer towards the end of the epidemic, when we finally received funding for more non-human primates. So we set out this study and did groups of 4 animals that we vaccinated with 10 to the 7th or 10 to the 6th PFU, and then we diluted it down to 10 to the 4th or 100 or even 10, 1, and 0.1. And you'll see in the data that that's also why we have a lot more control animals. In the first study, we did up to 100 PFU, not expecting that we would see a survival. However, it became necessary to do these lower dilutions. So we added control animals and ended up with 6 in this group. The vaccine was administered intramuscularly 28 days prior to lethal challenge. And what we found was really quite unexpected, but super fantastic, because at least in non-human primates, a dose of 10 plaque-forming units, which is a 1 in a million dilution to the dose that we normally give, was uniformly protective within 28 days. So if you think about, there's a vaccine shortage for an outbreak in Africa, you can definitely dilute it and not have to give 10 to the 7 PFU. That is great news. Of course, clinical trials now need to start to verify this, but Merck is on board in doing that. So this was really super exciting. And what we saw, that we even gave 1 PFU of the dose, 1 of the 4 animals in the group succumbed. And when we titrated it down to 0.1 PFU, I basically used 1 milliliter out of the 10 mils that we had in this group and diluted it with 9 milliliters of vehicle. And I guess I must have gotten that 1 PFU and into 1 animal, because here 1 animal lived out of the 4. So it was quite surprising. You can see here on the clinical score of the animals, they are averaged per group, that the animals, if they have something around or under 10, they're usually fine. They're just not eating very well, which is also caused by the anesthesia that they get every other day. That only the animals in the groups for the controls in black, for the lowest dose 0.1 PFU, and then for the 1 PFU dose got sick, but the others all pulled through without a lot of clinical signs. So that was super exciting. The animals, only the animals that actually were euthanized had developed viremia that went over our threshold of 10 to the 5th TCID 50 per milliliter in the blood. And only these ones also showed the characteristic drop in platelets and developed thrombocytopenia. So all of this is a typical picture of Ebola virus disease in these animals. When we looked by PCR, so because we wanted to know if the virus was replicating without us being able to titer it, we found that even in the group that received 10 to the 4th PFU, some of the animals on day 6 had virus replicating in the blood. And then in the lower doses, also almost all the animals at some time were viremic. And then only the ones, again, that crossed the 10 to the 5th threshold succumbed to disease. So this has been a very well-established euthanasia criteria for us in the lab now, and has been working really well, because all the animals are always euthanized. Nobody can go to endpoint death. Another surprise that came with this study was that the antigen-specific IgG response is not dose-dependent. I thought for sure if we give 10 to the 7 PFU, we will get much higher antibody response compared to 10 or 1 PFU, but that was not the case. You can see here in this ELISA data that 4 weeks after the vaccination on day 0, the animals had varying titers of IgG against Ebola glycoprotein. So that was super surprising. In this group, the one that seroconverted is actually the one that survived. So as seen with other studies before, in all the animals that survived, the Ebola virus challenge served as a boost to the antibody response to the Ebola glycoprotein. And you can see that that's always developing between day 6 and 9. It's a very nice, robust boost that then weans down a little bit over time, as long as we keep the animals until study at endpoint day 42. When we looked at neutralizing antibodies, as seen in the past, they're also not a big player in protection, because on day 0 here, they are barely around the limit of detection, which is indicated by this line. However, at the study endpoint, day 42, all of them are quite high up there. So it's really interesting for us to examine this and do this. And then this was published in 2019, and we did a follow-up study where we wanted to know if the 10, when we shorten the time between vaccination and challenge again to 10 days, which was seen in the human clinical trial in Guinea to be the short time for protection, what dose we could use there. And we found that 10 to the 4th with our laboratory derived VSV Ebola vaccine resulted in 50% survival. However, if we used the GMP vaccine that is actually for human use, then we get 100% survival. But we haven't dosed it down any further, and I don't think we will, because this is already a 1 in 1,000 dilution of the original dose, and I think that's good enough. If you can get 1,000 doses out of 1 vial instead of 10, I think that's awesome. So here again in this study, we only saw the virus load in the animals that succumbed, that climbed up over time, past the threshold of 10 to the 5th. And then also in the lab, we had developed an Ebola virus soluble glycoprotein specific ELISA, and that is detecting the second protein that expressed from the Ebola virus open reading frame. And that is only also detected in animals that really are highly viremic and develop disease and succumb. So that was a really nice correlation, so that now we can not just look for virus in the blood, but also for one viral protein by ELISA. So that's really cool. The study was finished about a year ago, and then we couldn't work on it anymore because everything was stopped. We needed to work on COVID-19. And we did that because, of course, we had no other choice. However, what we did is we used this VSV Ebola vaccine as our backbone. And I'm going here, because I've been talking too much, going to skip a few slides and just go into this project, because this was really the groundbreaking work that we did in order to make the VSV COVID-19 vaccine. So a postbac and a postdoc in the lab worked together on this. And what we wanted to do was using the VSV Ebola vector, since it was approved for human use, as a base vector and then add more antigens in. And you can see here, they made a whole bunch of constructs expressing the soluble H5N1 or a full-length version of it. So this is the full-length version of HA, the influenza hemagglutinin. And then they made a soluble version of it because it has been described that this is actually pretty good as a vaccination antigen in the literature. And this virus, the VSV, as long as it has the Ebola glycoprotein, it does not need a second glycoprotein for replication. So it was perfectly fine to put the soluble glycoprotein that is secreted from the infected cells into this vector. So we did this, and what we found was that, first of all, they were really immunogenic. However, we saw a difference. Only the ones that had the full-length glycoprotein resulted in robust seroconversion, whereas the ones that had the soluble ones did not. And that was a surprise because that's not how it was described in the other papers introducing these soluble HAs as antigens. Nevertheless, we moved on and performed an efficacy study in mice. We used the HA from the Vietnam strain that we have in the laboratory that we can also use as challenge strain. And we vaccinated the animals 4 weeks before challenge, and then challenged with 100 LD50 intranasally on day 0. We did a single dose of the vaccine or a prime boost, and that was the vaccine was given intramuscularly, not intranasally. And we found that only all the vaccines that had the soluble HA, the animals lost weight and some of them succumbed to disease, whereas everything that had the full-length HA by itself or together with the Ebola glycoprotein survived. So that was really good. We also did a time course and found that we can vaccinate up to a week before, and with this vector as well as with this one expressing the HA full length, the mice all lived after challenge. Then we submitted this paper and we're super excited, but the reviewers wanted to know if there's also protection against heterologous H5N1 isolates. And thanks to our collaborators from University in Wisconsin, Yoshikawa Oka, we got a hold of 3 different H5N1 isolates that we can work with in the BSF4. They are from different clades than ours. And when we vaccinated intramuscularly with a single dose and challenged mice 28 days later, we found that for both vaccines expressing either the Ebola glycoprotein and the hemagglutinin full length or the hemagglutinin alone, all the mice lived, no matter if they were challenged with an isolate from Anhui or Vietnam, from ducks, or from Indonesia. So that was super exciting because even though it was an IM vaccination, we are protecting against a respiratory virus. And that really was for us the headline to go ahead and try and develop a VSV against SARS-CoV-2, even though normally we work with diseases and viruses that do not necessarily cause a respiratory disease. So this was the ground laying work for that. And I don't probably have to tell you all about SARS-CoV-2 or the spike protein, but here is just a little cartoon what it looks like. And of course, this was our antigen of choice. And we made 2 different vaccines. One was based on the VSV Ebola, where we put in the SARS-S full length that was codon optimized for human expression. So we get these virus particles that have the SARS-S as well as the glycoprotein on the surface, or we just deleted the Ebola glycoprotein and put in a SARS-S by itself that had a 21 amino acid deletion at its cytoplasmic tail, because otherwise the virus wouldn't replicate. So we made these, recovered these, and then did Western blotting and could confirm that there is really good antigen expression. We see the SARS spike protein here in both the single vector as well as the bivalent with the Ebola glycoprotein, and we see the Ebola glycoprotein in the 2 vectors as well. A first clue that there might be a difference in protection came from growth kinetics data, when we infected Vero A6 cells at an MOI of 0.1 and found that the vector in red, having the SARS-CoV-2S as well as the Ebola glycoprotein, replicates similarly to its parent VSV Ebola, but the one that is only carrying the SARS-2 glycoprotein spike was significantly lower in endpoint titer, and you can also see that it's coming up later. So that was the first clue we had that there's definitely a difference in replication kinetic, and that this could also, in the end, be a problem for potential vaccine, because we know from Merck that the VSV Ebola is working well and can be produced and manufactured well. But if you have something that's 2 log lower, it might be a different story. So we first set out and tested this vaccine in hamsters. And because other people had already described that the VSV with the SARS alone works at 28 days in hamsters, we thought, okay, in a pandemic, it's probably really good to have a vaccine single dose that works fast. So instead of waiting 28 days, we vaccinated hamsters only 10 days before we gave them a big dose of SARS-CoV-2. So we tested the SARS-2 vaccine, the one that expresses SARS-2 spike and Ebola glycoprotein, and we use the Ebola vaccine as the negative control vector. We vaccinated them intranasally as well as intramuscularly to see if there is a difference. Then 10 days later, we challenged with 10 to the 5th TCID50 of SARS-CoV-2, the Washington 1 strain that probably a lot of you are familiar with and are working with. And we gave a rather high dose of 10 to the 5th TCID50. This model is not lethal, as you probably also all know. And so we just decided, in order to have a readout for protection, to euthanize all the animals 4 days after challenge. And we collected lung samples. We did lung cross-pathology pictures, and you can see here that the VSV SARS-2 that expresses this S protein alone by the intramuscular route did not protect from disease. However, when it's given intranasally, it does. In contrast, the VSV SARS-2 with the Ebola virus glycoprotein protects from disease by either route. So that was super good news. And then the parental vector, the negative control, the animals developed pretty severe disease, as so shown here by lung pathology. We actually just euthanized this morning some animals that were vaccinated intranasally with these 2 vaccines and challenged with the UK strain, and we found the same result. So this vaccine does not just protect against the Washington strain, but also against UK. We are having animals sitting that are vaccinated to also challenge with the South African strain next week. So hopefully that works as well. And then this vaccine will be a really good option because it can achieve cross-variant protection. When we looked by histopathology into the lung samples that we took from these hamsters, you can see in the negative control where they got the VSV Ebola either intranasally or intramuscularly, there was quite, some lung lesions developing with immune cell infiltration and thickening of the endothelia. And then, of course, lots of antigen expression here by the brown staining specific to SARS-CoV-2. The VSV SARS-2 vaccine that is expressing only the spike protein intranasally, vaccinated as well as intramuscularly, we saw a difference. Intranasally protected from disease, as was evidenced by the gross pathology pictures. And then here intramuscularly, we saw a lesion. However, there is no antigen present, so that was very good news, very different from the controls. The best outcome had the animals that received the vaccine with the SARS-2S as well as the Ebola virus glycoprotein by the intranasal and the intramuscular route. And here we didn't see any lung lesions nor antigen expression. So that was very, very good. And we were happy to see this. We, of course, do not know yet what it will be looking like with the UK and the South African strains, but hopefully this data is only a few weeks out. When we looked at viral RNA in the lungs of these animals, we found no difference in the nasal swabs. So even though the animals were protected from disease, there was still viral RNA in the nasal swabs. So transmission of the virus is probably still going to happen. However, there was a significant reduction in SARS coronavirus 2 RNA in the lungs of the SARS-2 EBOV as well as the SARS-2 vaccinated hamsters in red and in blue here compared to control. I really have to say that the hamsters have been the best model for SARS-CoV-2 to work with, because as you can see here, the model is so well established and so consistent, like we get almost no error bars in groups of 6. So this is really it's really nice to work with. When we looked at antibody responses, we saw that in the serum from 4 days post-challenge, which is also 14 days after vaccination, we see a significant increase in their SARS-S specific IgG, as well as in their SARS-S receptor binding domain specific IgG, and that the hamsters with the dual vaccine are significantly higher in these antibody responses compared to the ones that got the spike alone. When we look at neutralization, we see a similar picture. And interestingly, also there is not a significant difference between intranasally and intramuscularly when we look at the dual vaccine. So after these encouraging data, we decided to use a cohort of 16 rhesus macaques that we had on campus and I could get a hold of. And we performed a confirmatory study in the rhesus macaque model. We divided them up into 2 groups. One was intramuscularly vaccinated with the vaccine expressing SARS-CoV-2S and Ebola GP, and intranasally with the same. 2 animals in each of the cohorts received the parental vector, and they were then later lumped together as our control group of an N of 4 versus we had an IM study group of 6 and an IN study group also of 6. But it was really not justifiable to use more controls. So we vaccinated with a single dose standard 10 to the 7th PFU by either intramuscular or intranasal route, and then challenged 10 days later on day 0 with SARS-CoV-2 by intranasal, intratracheal, oral, and ocular installation, as was developed here at RML by Emmy DeWitt and Vincent Munster. We performed bronchoalveolar lavage on day 3 and took a sample. And then we euthanized all the animals on day 7, because again, this is not a lethal model, unlike Ebola virus. And we used the tissue readouts for viremia as our, basically readout for a protective efficacy or not. Again, when we looked at lung gross pathology, we saw clean lungs in the animals that were intramuscularly vaccinated, depicted here. The control animals developed lesions. To our big surprise, the intranasally vaccinated animals developed lesions similar to controls, or even worse than controls. And here that one control animal didn't develop lesions. But that is just the nature of the model, because not every control animal develops lesion that we have seen so far. When we looked by histopathology, we can again see here in the control animals indication of interstitial pneumonia, no indication of that in the VSV intramuscularly vaccinated animals, however, quite some pneumonia in the intranasally vaccinated animals. When we then focus in, we can see thickening of the alveolar spaces, they become smaller. In the control animals, only around a blood vessel here in the intramuscular group, whereas in the intranasal group, this is really much worse. And there's also a lot of immune cell infiltration. When we looked for antigen staining by immunohistochemistry, we saw spotted antigen, as has been described before, in the control animals and intranasally vaccinated, but not in the intramuscularly vaccinated animals. Next, of course, we looked for viral RNA in the samples that we took. First of all, in the nasal swabs, and we looked here for whole genomic RNA as well as subgenomic RNA, which is really indicative of virus replication. And in the nasal swab, there was only a significant difference early on between intranasally and intramuscularly vaccinated animals on day 1 and day 3, and to controls. However, in the subgenomic RNA indicative of virus replication, there was only a significant difference in day 1, and then it leveled out. When we looked at virus RNA, total RNA, and subgenomic in the BAL and ABAL on day 3, I for sure thought that intranasally this would look better. Well, I was so wrong. There was only a significant decrease in viral RNA in the intermuscular group, but not in the intranasal in red. So that was a big surprise, and it was observed in both of these with the total RNA as well as with the subgenomic RNA. When we looked at the lung radiograph scores, our veterinarians couldn't see any difference until day 7 when we euthanized the animals, when there was no indication of lung infiltrates anymore in the intermuscularly vaccinated animals. However, there was some in the control and intranasally vaccinated ones. When we performed RNA analysis on the lung samples that we took, and here they're lumped together, we took a sample from each lung lobe. You can see that again, we observed only a significant difference in the intermuscularly vaccinated animals and not in the intranasally compared to controls in black. So this was encouraging and also surprising, but then we wanted to know, okay, so what is it? Do we have less antibodies in the intranasal group, or what's going on? And we performed the same ELISA neutralization assay as I just showed with the hamsters. And we could see that indeed the IgG specific for SARS-S here on the left is significantly higher at the time of challenge, which is 10 days after vaccination, only in the intramuscular group compared to intranasal or controls. Then the intranasal is catching up, but it's always significant except for day 1. The difference stays that way. When we look at receptor binding domain specific IgG, we see a similar picture. However, that becomes not significant from day 1 on throughout the study, even though the intramuscularly animals seem to be slightly higher in IgG specific to the receptor binding domain compared to the others. And in neutralization, surprise, surprise, there was no difference between the 2 vaccinated groups. So it seems that even though antibody quantity is significantly different here by ELISA, quality when it comes to neutralization was not. How this all plays together with, why intranasally was also showing those lesions and intramuscularly not, we have addressed with Ilham doing transcriptomic analysis, but I wasn't too confident showing those data, not to her, to all of you. So if you have questions, I'm sure she will be happy to share them because they're quite interesting. We looked, however, at antibody responses in the BAL, and we found only that there was one animal that had quite high SARS-S specific IgG. There was nothing really for receptor binding domain or even IgA. And that was a big surprise because we were expecting to see, particularly in the intranasal group, some S-specific IgA in the lungs, but that was not the case. When we looked at IgG subtypes, we found that the response after vaccination, here with the empty columns, was IgG1 biased, and that got boosted. And interestingly, the IgG2, the little that we had in the serum, disappeared after challenge within the week, and IgG3 was boosted. In the BAL, there was the little antibody response that we had was IgG2 or 3, no IgG1. So that's also different. And lastly, the virus specific IgG was very similar between control as well as intramuscularly vaccinated animals. which would indicate that would we do an Ebola virus challenge with this vaccine if the development of a dual vaccine was really our premise, that would probably have been achieved. But of course, we would have to actually do the study in order to be 100% certain. But from what we know and the antibody levels we see to the Ebola glycoprotein here, they should be protected. So last, I'd like to summarize and go a little bit into ongoing work. I've shown you our data that we have developed a VSV SARS-2 vaccine based on the VSV Ebola virus vector that's approved for human use, and that has performed superior to the Merck vaccine that was actually just a month ago pulled from the market, or like development was stopped. It wasn't even on the market yet. Our vaccine works within 10 days in hamsters and non-human primates, and to our surprise, intramuscular vaccination was more efficacious than intranasal in non-human primates. As I said, Ilham's team, particularly Amanda and Ellen, did transcriptional analysis of bell and lung samples, and it revealed the activation of innate and adaptive immune responses in the intramuscular vaccinated animals only. But I hope she can elaborate on that should you have more questions. We are right now in the updating the vaccine vector, including spike variants from those new strains from the UK and South Africa. We are also running neutralization assays against the serum samples we have from our studies. We are performing efficacy studies in hamsters, as I said, and we are also looking in the impact of pre-existing immunity to the VSV Ebola vector on the protective efficacy of all those bivalent vaccines that we have developed. I would like to thank everybody that was involved. Animal studies always need a lot of help and support, and here at RML we really have that. We have a great crew in the veterinary branch, veterinarians, pathologists, as well as animal caretakers, so those deserve all the pat on their shoulders. They're awesome to work with. And then I'd like to thank particularly my lab, Waka, who made the vaccine, Kyle, who took all the stuff on with the hamsters, and Kyle and Talisha, who did a lot of work on the RNA stuff, making standards, running the ELISAs. Like, they were tremendously helpful. And then everybody else who was involved, and again, my collaborators at UC Irvine, Ilham and Ilham's team. And then lastly, should you be a grad student finishing up and looking for a postdoc and maybe wanting to work in a BSF4 at some point, let me know. I have an open position starting this summer. So if you're interested, seriously, really just shoot me an email and we can chat. Thank you. I'm happy to take any questions you might have.
Well, thank you, Andrea, for that absolutely wonderful, wonderful talk. There are actually a number of questions in the chat box. So what I'll just do, I'll just call upon people to unmute and show their video, and they can ask their questions. And so Paul asked a provocative question, which actually we did in our journal club and M1 in our virology class for undergrads. So 0.1 PFU group, we actually had some protection. How does that work? And I can give you what I came up with for the students, but I'd love to hear your interpretation. I think Paul wants to know how that all works too.
Well, yeah, I had no idea. So the back titration came back negative. There's no virus in there. But as I said, I made 10 milliliters of my 10 PFU and then used 1 milliliter out of that and put it in my vial with 9 milliliters of diluent. I might have gotten lucky and gotten that 1 PFU and gave it to that monkey. I don't know. That's really the only explanation we have.
When you determine the original titer of the virus, did you try several different cell lines? Did you spinfect?
No. No, it's really with VSV, it's such a beautiful virus to work with. You just infect cells as plaques. You can do regular agarose overlay. It's very beautiful. We always work with OE6 in low passage. So I don't know if, but that's our standard cell line we work with all our viruses with, not just with the VSVs.
I see.
Sorry,
But
Paul, go ahead.
there's a better question further down, which
Oh,
somebody,
okay.
yeah.
No, go for it. I was, can
Well,
I,
I mean,
can I ask
somebody else.
Andrea for, what are your thoughts on defective particles acting as a vaccine in this case?
It could be, and normally we have some in the prep, but not as many as you would think. You know, like when we worked with the filoviruses, we actually have this virusite counter here, that we have an idea that about like filoviruses, we have one real particle and about 1,000 defective. With the VSVs, since we harvest them much earlier than the others, we don't. So we think maybe it's a 1 in 10 or so at the most. So that is definitely a possibility, yes. But it was interesting that in that group, though, only that one animal that was protected had an immune response, and the other 3 did not. So it's like that one got lucky. It must have gotten whatever was in there in my 10 mils. I don't know.
Paul, I think, sorry, we were thinking alike. That Bert has a question, so I'll just try to be quiet.
I just wanted to know if the protected non-human primates that exhibited the low levels of viremia, are they actually capable of transmitting the virus?
For the
Ebola.
For Ebola, we don't know. So as long as we can, for the ones where we can isolate the virus out of the blood, which is usually more than 5,000 TCID, 500 TCID, 500 genome equivalent, then if we can isolate virus, then yes. If it's below that threshold, then we can just detect RNA. I don't think so.
Because I have this concern that, you know, you were talking about in Africa diluting out the virus a lot. So if you get some version of protection, but not enough to actually stop virus replication, then you might still get transmission. And so that might be a balance that you want to stay away from in terms of getting too diluted of a virus vaccine.
Yeah, very good point. Yes, definitely.
Thank you.
And okay, Lisa, and then I'll come back to the person above Lisa because I don't know who they are. But Lisa, do you want to ask your question?
Sure,
That person, Ilham, is Eric Stanbridge.
Oh, okay, good. Thank you. I'll let Lisa ask her question. I'll go back to Eric.
I'm just interested in your flu results. So with the H5 data, how you had the soluble and the full length forms of HA. Do you know what's going on with the soluble form? Are you making full trimers? Are they folded correctly? Like, do you think you're getting different kinds of antibodies that are maybe not protective?
Exactly. So we had 2 versions in there, and because of the interest of time, I didn't go in it. We had just a soluble HA, and then we had one that was also having an isoleucine zipper to stabilize the trimer. But we were never really able to fully determine, even by microscopy effort, what we really got, or by native gels. So that was a bummer. And we have not looked into the quality of the antibodies after this, but that is definitely something that is a possibility that could happen. Yeah, that was, of course, forefront of our mind. That's why we made the ones with the stabilizing domain. Yeah, most definitely.
Great, thanks.
All right, Eric.
Yes, Ilham, I think you've already asked that question, and it was answered by Dr. Marzi after you were following up from Paul Gershon. And it was the issue of the number of pseudovirions that might be possible, present in the PFU assay. Right. And as you correctly pointed out, with some viruses, it can be a huge ratio difference. But as you indicated, I think it's not such a problem with VSV. Is that so?
That is correct. We see it some, but not as badly as we see with others. Like we have, for example, a huge issue with that for Lassa virus, but it's all different.
Yeah, although I must say, as you go into these hybrid constructs now, Ebola and VSV, et cetera, that may change that ratio somewhat. And I think it would be worthwhile to check in all of those quality controls that you do, that the pseudovirion thing doesn't lead you astray with some of those.
Yeah, that's definitely a good suggestion. And we might do that because we have the virus site counter on campus. So and VSV is BSL2, so that would be very easy to do.
Okay.
Okay, we're a little bit over, so maybe a couple of last two quick questions. Alexis, do you want to go ahead and ask your question?
Okay. Yeah. So I've seen that for your VSV SARS-2, you had a deletion of 20 nucleotide at the end on C-term. So do you think it could be the reason why you have a differential protection with your Ebola SARS-2, or is it just the bad replication that just not producing enough proteins?
It could be that those 21 amino acids, because they are really in the entire cytoplasmic tail, would have some interaction intracellularly if they get expressed from an immune cell. That's definitely not to exclude. However, if we leave them there in the VSV vector by itself, it won't grow. Like there is something encoded in those 21 amino acids that's impairing the incorporation of the SARS spike glycoprotein into the VSV particle. So we had no other choice than to delete it. And actually Sean Whelan's group, as well as Kartik Chandran's, they also described this, and they had either 21 or 19 amino acid deletion. So it's very intriguing. I was actually surprised that it would work because, you know, it's a completely different virus. It's positive strand, single strand, but yeah.
I wonder what Merck did. We will never know.
No.
And then Dr. Buckmeyer, I leave the last question to you.
Okay, I'll just take one of my two. The second one I think you've answered. In a nutshell, what is your current view of the role of the soluble S protein in pathogenesis?
Soluble S for Ebola?
Well, for Ebola, yeah. It's been argued for years and years and years, and nobody's ever come to a conclusion.
Okay, so my postdoc, who just started as assistant professor this week in Japan, actually did a whole bunch of stuff on that. And we found that it is a contributor of pathogenicity, and it is activating the MAP kinase pathway. So even when you just put the soluble glycoprotein at different doses onto cells without the virus, you get activation of the MAP kinase pathway. And then, so it is contributing to pathogenicity. She is actually taking this project with her and diving into it further, because this is something she can do in BSL-2. But what we did, we went ahead here and did a very cool mouse study where we infected wild-type mice with two viruses, one wild-type expressing the soluble glycoprotein, and the other one where we made a mutation that it's not expressing the soluble glycoprotein anymore. And wild-type mice without mouse adapted do not succumb to disease. However, the virus replicates. And then after a few days, we stimulated these mice with SGP or with PMA or with just control protein. And we found that virus replication, particularly in the liver, went up with SGP and with PMA, both. So that has been, another indicator that there's something more going on. So we are definitely thinking it is contributing to pathogenicity. By which mechanism? MAP kinase, but there's so many options still that we need to check out. So it's in the works.
Thank you.
Well, thank you again for giving us such a wonderful presentation and talk. The students will stay on, and then the rest of the faculty will trickle in. But it's all the same Zoom link, so I think you don't have to go anywhere.
Okay, perfect. Really I'm just going to stay put.
Yeah, you just stay put, and some of us will come in and out of your life for the next few hours.
Thank you. Thanks all. It was a pleasure. Thank you.
Awesome. I'll see you at 4. Bye.
Bye.